This machine may hold the secrets to time. Inside it, scientists are looking for the mysterious mechanism that controls the daily biological clock.
Everything in your body is controlled on this 24-hour process.
The things that seems most interesting to me about circadian biology is the fact that it spans many timescales and many size scales.
For more than a quarter of a century, chronobiologist Dr. Carrie Partch has been peering inside our biological clock mechanisms, trying to understand just what makes them tick.
I was hooked. As soon as I began graduate school, I really knew that I wanted to work to unravel the molecular details of circadian timing.
In 2020, Dr. Partch was diagnosed with the neurodegenerative disease, ALS, also known as Lou Gehrig's disease.
Due to ALS, my speech is beginning to decline, so I use an AI clone of my voice to give talks and answer questions like this.
But she's still in search of answers to how the clocks in our cells govern our days and nights.
I believe we all need a purpose. I can't imagine not working. Also, I really enjoy the moment of discovery in research. After putting in lots of thought and hard work, to be awarded with a fundamental insight into life is about as good as it gets.
Chronobiologists study the effect of time on biological cycles. Those could be as fast as a heartbeat or as slow as a seasonal animal migration. There's one rhythm most of us know intimately, the circadian rhythm, which governs a living organism's 24-hour clock. This daily synchronization was first observed in the early 1700s when a French astronomer put a plant in a dark room and watched it respond to the cycles of day and night. Since then, researchers have been trying to decipher just what sets the rhythm of these clocks. Advances in DNA sequencing in the 1990s led to breakthroughs that began to unravel the mystery by identifying the genes that govern circadian rhythms. In 2017, a trio of researchers were awarded the Nobel Prize for their work on the molecular mechanisms controlling fruit fly circadian rhythms.
Identifying the genes required for timekeeping and understanding how they work together in a genetic network was an incredible step forward for our field.
That was the end of the story for many scientists, but not for all.
For people like Carrie and me and other people in structural biology, there is another layer that I think we really want to know.
I'm really interested in the molecular steps of these biological clocks and how protein-based signals in our cells can measure out a day.
The circadian clock is triggered by the sun, but it's not just about sleeping and waking. The circadian clock also governs more than 40% of human genes, which regulate myriad other essential functions, including metabolism, hormone production, and body temperature over the course of the day. In fact, some medicines, like acetaminophen, are better metabolized in the daytime, and vaccines can work differently depending on when they are given in the morning or at night. Many aspects of modern life, like traveling across time zones and exposure to artificial light at night, can wreak havoc on our natural circadian clocks. But being out of sync with the natural cycles can cause more than just fatigue. Working the overnight shift is associated with diabetes and heart disease, as well as certain cancers. To fix this mismatch between our bodies and the modern world, Partch is searching for ways to tweak the circadian clock. But first, she had to map it.
For us to understand sort of how the gears of the clock turn, we need to understand how the proteins find one another, how they interact, how long they stay together, and how that might be changed over time.
To study the human biological clock at the molecular level, Partch's lab uses a technique called Nuclear Magnetic Resonance Spectroscopy, or NMR.
NMR is very highly related to MRI, which many people will know a lot more about. What NMR and MRI have in common is we both need really big magnets.
In an NMR tank, liquid nitrogen and helium cool a superconducting magnet, which produces a magnetic field 1,000,000 times stronger than Earth's.
So why do we do all that? That high magnetic field lets us be able to detect signals coming off of many of the atoms inside of the proteins or drugs or DNA or RNA that we're putting inside of a tube in the middle of the magnet.
To study the biochemical and biophysical mechanism of the circadian rhythms, we need to make proteins outside the human body in test tubes. We take the DNA and we tell bacteria to do that for us. So this is the NMR tube. We put it in the magnet to take the reading.
Using NMR, researchers like Partch can see a picture of proteins at work.
NMR is sort of like taking a series of GPS snapshots of a protein. NMR gives us information about the environment around certain atoms in the protein and how they move, which lets us infer how the protein wiggles around in solution and responds to its partners, like other proteins or drugs.
Real power of it is that all the work we do is literally on samples in solution at room temperature.
In Partch's lab, the samples are often of what's called clock proteins.
We call the proteins that interact with each other and our genes to regulate our circadian rhythms clock proteins, just like how the gears of a mechanical clock work together.
At the heart of the process are two protein molecules called clock and bmol. Each day at dawn, clock and bmol pair up in the cell's cytoplasm before entering the nucleus. Once inside, they bind to the DNA on thousands of sites to regulate the production of proteins important for daytime functions. The clock-bmol pair also regulates the production of two repressor proteins, period and cry, which play a critical role in their circadian feedback loop. Levels of period and cry build up during the day. Then at night, they enter the nucleus to strip clock-bmol from the DNA, shutting things down. Over the course of the twilight hours, the repressor proteins decay, diminishing their effect, while the supply of clock and bmol proteins builds back up. In the morning, the whole cycle begins again. However, genetic variations can alter this protein clock cycle, leaving some out of sync with the day and night light cycles of Earth.
One tiny change in a clock gene, an inherited change that only alters a few atoms in the protein it encodes, can dramatically alter your internal clock timing and change when you go to bed and wake up. I'm sure we all know some morning larks, folks who go to bed early and enjoy getting up with the sun, or night owls who like staying up late. Well, some genetic changes can change clock timing by hours, leading to bedtimes at 5 p.m. or 4 a.m.
By studying the smallest components of our biological clocks, Partch's lab is searching for ways to correct problems caused by genetics or the intrusion of the modern world on our circadian rhythms.
We're using NMR now to discover and map how small molecules bind to clock proteins, with the hope of turning these into drugs that could help people adjust to shift work or reduce jet lag, or even treat certain cancers like glioblastoma that depend on circadian machinery.
To Partch, the world is full of clocks waiting to be pulled apart and understood.
Oh, there is still so much more to learn about our own clocks and circadian rhythms in other species, like plants, that could be important for sustaining life on Earth and beyond. It's exciting to see an awareness of circadian biology and its impacts on our health finally begin to grow in the clinic. Hopefully, we can build on this to incorporate a better understanding of how time influences our biology and improve everything from our sleep to how we respond to drugs.